Welcome everyone to my talk on leveraging the interactions between plants and soil for sustainable crop production. First, I will share with you my overarching research goal and my motivation behind it. Then, to become more concrete, I will tell you about some of my previous research. And after that, I will provide some insight into what my PhD students are currently planning to work on these coming years. So I think we all agree that one of the pressing global challenges is to figure out how to meet the increasing demand for food while reducing the negative environmental footprint of agriculture. And further, I think we agree that there are different directions from which to approach this question. Agriculture occupies almost 40% of the Earth's ice-free terrestrial surface, and this map shows the world's croplands yield that is gained relative to the potential yield that could be achieved in any given area. The yellow shows high-yielding areas, and the orange and red show areas where yields could potentially be much higher than they currently are. In sub-Saharan Africa, you see that there are extensive areas where yields could be much higher than they currently are. And this might be due to various factors, one of them being that the majority of farmers in sub-Saharan Africa lacks access to sufficient external inputs, such as fertilizer and pesticides. This map also nicely illustrates that croplands in Western Europe and North America are mostly high-yielding. In these areas, external inputs tend to be highly used, and while allowing to generate high yields, it comes at a high environmental cost. Agriculture accounts for 25% of all greenhouse gas emissions worldwide. It is responsible for the loading of nutrients, especially nitrogen and phosphorus, into the biosphere and pollution due to pesticides. And agriculture is the single largest cause of biodiversity loss. Further, agriculture consumes 70 to 90% of all fresh water that is used by humans. And yet, there are 800 million people hungry worldwide. So if we are to help achieve the United Nations Sustainable Development Goals, such as zero hunger and life on land, the challenge is to find a way to increase and or maintain yields without extensive use of external inputs. What could provide at least part of the solution, what could enhance the sustainability of crop production across farming systems worldwide, is if we better understood how to best harness ecological processes. So leveraging ecological processes and specifically the interactions between plants and soil abiotic and biotic variables to help achieve sustainable agroecosystems forms the foundation to my research approach. It's the direction that I have been and intend to continue taking to address the global challenge of meeting the increasing demand for food while reducing the negative environmental footprint of agriculture. My research approach is twofold. One, I take our fundamental knowledge gained from small-scale experiments into the field to enhance our mechanistic understanding of plant-soil interactions in real-world agricultural context. And two, by asking contextualized farmer-relevant research questions, I seek to better understand how to best design and manage agroecosystems for optimized functioning and high adoption potential. And I seek to take a holistic approach to sustainable crop production, and therefore I aim to figure out how to harness the critical role that soil microbes and plant-soil interactions play in nutrient cycling and crop nutrition, in soil water dynamics and crop tolerance to drought, as well as crop resistance to pests. The reason for why I would like to look at these three aspects to crop performance is that they are intricately linked and all determined by plant-soil interactions. And I see potential in identifying diversifying practices, fostering plant-soil interactions with synergistic effects on crop nutrition and resistance to perturbation. So to become more concrete about what I seek to further build upon, I will share with you some of the research that I conducted in the past. This first takes me to a low-input agroecosystem in sub-Saharan Africa. This photo shows the farming system. Maize fields of subsistence farmers in Malawi, farmers that have no steady source of income and are relying on their maize yields to feed the family. And the trees especially nitrogen-fixing phytoerbia, those that you can see in the photo, are commonly distributed throughout smaller farmers' fields. And that's because they are known for the positive impact on soil fertility. So with the trees in farmers' fields, we have what's called an agroforestry system. And while the positive effect of agroforestry trees on soil fertility has mostly been attributed to high-quality organic matter inputs, such as tree leaf litter input, I was curious to find out to what extent mycorrhizal fungi contribute to the enhancing effect of trees on soil fertility. Mycorrhizal fungi, or AMF, are key root symbionts that require plants' carbohydrates and in return provide plants with several benefits, such as enhanced access to nutrients, as well as greater tolerance to abiotic and biotic stressors. And let's focus on the role of mycorrhizal fungi in providing host plants with greater access to nutrients. So what's been known for quite some time is that the mycelia, or networks of fungal filaments, fungal hyphae, they basically increase the surface area of the host plant's root systems, and as such improve access to nutrients. So this is shown in the figure on the right. You see in the circle on the right, the orange lines representing the mycorrhizal mycelia, networks of fungal filaments that can reach into soil pores that are too small for the roots to access. And this is in contrast to a plant with a root system that doesn't associate with mycorrhizal fungi. Here you can see on the circle on the left, the roots only have access to the areas of the soil that the roots can actually get to. More recently, we gained insight into the potential of mycorrhizal fungi to connect different plants via networks of fungal filaments, via mycelia, and transfer nutrients from one plant to another. And this could have potential in enhancing sustainability of low-input farming systems. For instance, if nutrients were transferred from well-established trees, such as mango and phytoerbia trees that you can see in the photo, to surrounding crops, such as maize. However, most of our knowledge about mycorrhizal fungi and interplant nutrient transfer is limited to simplified and small-scale pod experiments. Knowledge on whether these findings can be translated to complex systems is still scarce. I set up a root exclusion experiment around single-standing phytoerbia trees in farmers' fields, and I used isotope tracing to test the hypothesis that mycorrhizal fungi allow maize to gain access to tree-derived nitrogen that is otherwise inaccessible to maize. The photo in the bottom shows the experimental setup, which allowed us to distinguish the different mechanisms underpinning nitrogen uptake by maize. We dug out these plots, 1 meter by 5 meters and half a meter deep, put in place different types of lining, then put the soil back in place, let the system rest for a year, and then we sow maize and sand maize throughout the plots to determine the 15N isotopic signature. And for the sake of time, just briefly, the 15N isotopic signature of the nitrogen-fixing trees is distinctly different and therefore allows distinguishing the tree-derived nitrogen from the soil-derived nitrogen. The figure shows on the x-axis the distance of maize plants from the tree. So we sampled at 1, 2, 3, 4, and 5 meters from the tree. On the y-axis, it's the proportion of tree-derived nitrogen in maize leaves. And the different colors represent the different mechanisms through which maize obtain the tree-derived nitrogen. So in green, it's the nitrogen that was derived via leaf litter input. In orange, via mycorrhizal mycelia that extend beyond the maize rooting zone. And in blue, via direct root-to-root contact between tree and maize. As you can see, most of the tree-derived nitrogen in maize is due to tree leaf litter input. The green line represents the average of the tree-derived nitrogen in maize due to litter inputs. What's remarkable, however, is that half of that, so about 1/3 of the total tree-derived nitrogen in maize leaves is due to mycorrhizal mycelia. Roots had basically no effect. So these findings further promote maintaining trees within fields, and additionally, they provide a mechanistic insight in that they suggest that managing for mycorrhizal fungi in farmers' fields can improve crop nutrition, which may result in greater yields. But what these findings didn't tell us was whether the uptake was mediated through a direct tree to maize nutrient transfer or tree to soil to maize, or in short, soil to maize transfer. And if it's the latter, if it's a soil to maize transfer, is it tree-maintained or maize-associated mycelia that take up the tree-derived nitrogen from the soil and transfer it to maize? I mentioned mycorrhizal fungi require plant carbohydrates, and so they do present a cost to the plant. And now the question is, do trees or maize seedlings cover the cost for these mycelia that allow a transfer of tree-derived nitrogen? So I conducted an isotope tracing experiment in the greenhouse. I grew mango and Phytoerbia trees within Malawian field soil, so within the native soil microbiota. And then I planted maize 2 years after I planted the trees into these ingrowth cores that prevented a root-to-root contact between the maize and the tree, but allowed for potential nutrient transfer via mycorrhizal network. Here's a schematic of the experimental design. So to test a tree to maize nutrient transfer, we applied 59 and 33P isotope labeling solution directly to the tree stems via stem injection. Again, just briefly, in this case, the 59 and 33P that we applied acted like a dye that we were able to trace into maize seedlings. To test the soil to maize transfer, we applied 59 and 33P isotopic labeling solution to root-free labeling compartments that were buried in the substrate. And to assess the role of tree-maintained mycelia versus maize-associated mycelia, we tested the soil to maize transfer with and without trees. Before I continue, I'd like to direct your attention to the two types of ingrowth cores. As mentioned, maize was grown in ingrowth cores to prevent nutrient transfer via direct root-to-root contact between tree and maize, but allow mycorrhizal mycelia to potentially provide maize with access to nutrients from beyond the ingrowth core. To control for any non-mycorrhizal mediated nutrient transfer, we included one ingrowth core that was rotated just before label application, and that was to break any potential hyphal connections between tree and maize. So let's first look at the results for tree to maize nutrient transfer. The x-axis shows the sampling day post-label application. So we sampled and analyzed maize leaves for 59 and 33P at 7, 14, and 28 days post-label application. And on the y-axis, you see the specific radioactivity of 33P. I'm only showing results for 33P, but it's very similar for 59 as well. The diamonds and circles represent maize grown with mango versus Phytoerbia. And the green shapes represent maize grown in rotated cores. So the green shapes show nutrient transfer that was not mediated by mycorrhizal fungi because we broke the hyphae by rotating the cores. The blue shapes, on the other hand, represent maize grown in static cores, and as such, show nutrient transfer mediated by mycorrhizal fungi. You can see that there's no difference in the amount of 33P obtained by maize grown in the rotated cores, the green, and the static cores. So these results provide no evidence for a tree to maize nutrient transfer mediated by mycorrhizal fungi. For the soil to maize nutrient transfer, the layout of the figure is basically the same, with the addition of the triangles representing maize grown without trees. And here we do see that maize in static cores, so the blue shapes, show greater 33P than those where mycorrhizal mediated transfer was not possible. You see the green shapes are all around 0. and it's only the blue shapes that show an increase in 33P in maize with time. However, there was no statistical difference in the amount of nutrients obtained by maize growing with and without trees. So clearly, maize seedlings effectively establish their own mycorrhizal mycelia that provide them with access to nutrients from outside their rooting zone. And so it's possible that maize seedlings simply rely on their own mycelia to obtain nutrients, even in the presence of already established tree-maintained mycorrhizal mycelia. To summarize, my findings show that regardless of whether it is maize-associated or tree-maintained mycorrhizal mycelia, or a combination of both, mycorrhizal fungi can improve nutrient uptake by crops, and as such, likely play a role in the positive effect of agroforestry trees on soil fertility and crop yield. Okay, so now let's move from the low-input agroforestry system in Malawi to an intensive horticultural production system in California. Intensively managed agricultural systems, such as shown in the photo, typically show a lower mycorrhizal abundance and diversity than natural systems. And there are two likely reasons for this. One, crops are well supplied with nutrients. And two, intensive soil management presents a high level of disturbance, breaking mycorrhizal networks. And both of these factors affect the functioning of the symbiosis. As soil health management has gained increasing attention, so have soil microbes and bioinoculants. Bioinoculants are products containing beneficial microbes known to promote plant growth. There has been curiosity as to whether or not inoculating crops with mycorrhizal fungi improves crop performance in these intensively managed agricultural systems. There have been contrasting results on the effect of inoculating crops with mycorrhizal fungi. Some studies show a positive, others show no effect. We tested whether the effectiveness of mycorrhizal inoculation of crop seedlings in improving yields would depend on soil management practices. We worked with tomatoes in this case, and we found no clear effect of inoculation. So this figure shows on the x-axis the 20 different fields that we worked on. On the y-axis, the difference in marketable yield between inoculated and non-inoculated plots. The data points above the line show a positive response to inoculation, and the data points below the line indicate a negative response to inoculation. You can see the points are all over the place. The different colors represent 6 different soil management practices. So we had different types of green waste and manure, as well as no organic matter amendment. So we found no difference, neither in yield nor in food quality, between inoculated and non-inoculated plots. And so to me, these results drive home the point that we can't achieve sustainable agriculture by simply relying on external inputs, whether it's bioinoculants, fertilizer, pesticides. Instead, we have to figure out how to best design and manage farming systems that support optimal plant-soil interactions. To harness plant-soil interactions, we have to better understand how to best condition the soil in a given system. So we know that a previous crop affects the soil differently and the subsequent crop differently than another crop. And there are overriding factors such as landscape structure and soil properties that further impact these plant-soil interactions, as well as plant-plant interactions. And this knowledge has already been used to develop suitable crop rotations, for example. However, what fulfills one purpose may defeat another. And of course, depending on the agroecosystem, we may have to adjust the way that we harness plant-soil interactions. In the simple maize-based agroforestry system, trees can, for instance, help maintain mycorrhizal networks in the soil during the non-cropping season and improve soil structure and therefore nutrient retention. In more intensive agricultural systems, we may, for example, promote the abundance of mycorrhizal fungi through the use of mycorrhizal cover crops and minimal soil disturbance. And this leads me to the work that my current PhD students plan to work on within the local agricultural system here in Germany. the focus will again be on the mycorrhizal symbiosis, but beyond determining the role of mycorrhizal fungi in crop nutrition, we will examine crop tolerance to water stress and insect herbivory. Why water stress and insect herbivory? Well, because clearly both are relevant when aiming to achieve sustainable crop production, but specifically because mycorrhizal fungi are known to affect both. With regard to water, studies have shown that mycorrhizal fungi affect water relations, such as stomatal regulation, in ways that may optimize the responsiveness of plants to low or variable soil moisture. Also, mycorrhizal fungi stimulate antioxidant enzyme activity, which alleviates damage caused by reactive oxygen species under drought stress. And this can then improve water use efficiency and recovery of plants post-drought. Further, by enhancing plants' access to nutrients, mycorrhizal fungi can maintain the physiological performance of plants, and this has indirect effects on water dynamics as it strengthens plants' ability to recover and or prevent severe damage of roots and the photosynthetic apparatus. With regard to insect herbivory, there are again indirect effects of mycorrhizal fungi. Enhanced access to nutrients leads to greater plant vitality, and as such could result in greater resistance of crops to insect herbivory. The establishment of the mycorrhizal symbiosis in itself may prime plant defenses. This is referred to as mycorrhiza-induced resistance. And what I find particularly intriguing is that there's evidence for mycorrhizal networks to transfer warning signals. It's been shown under controlled conditions that intact mycorrhizal mycelia transfer warning signals from one plant that is attacked by insects to neighboring plants that are not yet attacked by insects. And the warning signals cause neighboring receiver plants to boost up their immune system, making them less susceptible to insect herbivory. However, the caveat remains that intensive agricultural systems negatively affect mycorrhizal fungi and break mycorrhizal networks. In the local agricultural systems, cover crops that are typically grown prior to a main crop can present a viable option to manage for mycorrhizal networks. If the cover crop is a species that associates with mycorrhizal fungi, which are basically all but members of the Brassicaceae and Chenopodiaceae, then the soil will likely be well colonized by mycorrhizal networks. Intact mycorrhizal networks in the soil present the best source of mycorrhizal propagules, leading to the fastest root colonization of crops and subsequent benefits to the plant with regard to access to nutrients, water relations, and resistance to pests. So, from the perspective of the mycorrhizal symbiosis, to keep mycorrhizal networks in the soil undisturbed, there may be good reasons to promote no-till systems. So the first question that we will address this coming season is, do intact, undisturbed mycorrhizal mycelia established through cover crops and maintained by performing no-till improve the tolerance of the preceding main crop to water stress and insect herbivory compared to the tillage-disturbed mycorrhizal mycelia? We will start with explorative field experiments and later then combine it with mechanistic experiments to actually decipher the role that mycorrhizal networks may play in enhancing crop tolerance to stressors. Okay, I will leave it at this. More to come as we progress with this work. So now just a quick recap. With my research, I strive to better understand and learn how to manage plant-soil interactions for greater crop nutrition, tolerance to drought, and resilience to pests, and as such take a holistic approach to attain sustainable crop production. And altogether with my work, I aim to help work towards attaining food and nutrition security, as well as reducing the environmental footprint of agriculture. And with that, I'd like to acknowledge the people that were involved in the work that I have presented to you today. Mentors, colleagues, students, assistants, farmers. And I'd like to thank you for your attention. Happy to take any questions.